Hello, College Biology. This is another cell membrane video. This will be over passive transport, specifically facilitated diffusion. Now, the word facilitate means to make something easier. I think facile literally means easy or easier in French. Maybe. I don't know. Anyway, to facilitate is to make something easier, and facilitated diffusion is normal diffusion, particles from an area of high concentration to an area of low concentration using proteins. Proteins make it easier, and that's all there is to it. We've got two categories of proteins that make it easier. No, I want this up here. Yeah. Okay. So those two types of proteins will be a channel, or, as is illustrated by these three things here, carrier proteins. Channels are very simple. They are simply open. They are tunnels, and they restrict what can pass through based on size and polarity. But if it works for the channel, it'll pass through. And so they're real simple. Whatever these hexagons, probably glucose, they can pass through. But whatever these ovals, I don't know, maybe a monoglyceride, I don't know. They can't pass through, although a monoglyceride would be able to just pass through here. And it doesn't have a fatty acid tail, so it's not a monoglyceride. I don't know what this is. But it can't pass through this channel, because the channel lets, things pass through based on size and polarity, and they can just diffuse through, which means that right now they're diffusing this way because there's a higher concentration out here. But should there be a higher concentration down here, then they would diffuse the other way. Channels are bidirectional. Doesn't matter. Now, carrier proteins are not necessarily bidirectional. They bind to a specific thing based on polarity, which causes a conformational change in the shape of this protein, which then releases them. So let's take a closer look at protein channels right there. This is an aquaporin. I think we mentioned this in the osmosis discussion, and it allows water to pass through. And it's embedded in the cell membrane, and it's an open channel, and that's pretty much it. That's pretty much all there is to it. Of course, this is our cartoony representation. If we try to get into their shape more specifically, they look more like this. This is a bunch of alpha helices in this protein, and we have two different colors, which means we have two different subunits here. And so, that's what you get, an open channel. Let's take a look at a carrier protein. Here, we've got glucose. This is going to be a glucose carrier protein, and as it reads, the transport protein has a binding site for glucose. So the polarity of the glucose and the polarity of the protein attract each other. For glucose, that is open to the outside of the cell. So this is, again, a one-directional carrier protein. It comes in, binds, causes a conformational change. Polarity causes the protein to change its shape. And so then it can slip out and be released because the polarity changes, so they are no longer attracted to each other, so it'll wander out. And then this will cause renaturing because polarity will revert back to its original position. Carrier protein. And of course, carrier proteins don't really look like this. They're a big jumbled mess. This is a particular carrier protein that has so many subunits, because each one of those different colors is a different subunit. And you can see not only the alpha helices all over the place, but you can see these arrows representing parallel lines. Instead of drawing the actual parallel lines back and forth, they just show the arrows representing 2 or 3 foldings back and forth. But there you have it. That is facilitated diffusion. Diffusion with the use of proteins, and they can be either channels, which are just open to specific sizes and polarity, and carrier proteins, which are specific, which move specific things. I hope this helps. Struggle well.